And let's see, how do I advance slides? Do I have to go? There we go. Go back. So, hi everyone. First I want to check, I want to make sure that the audio is okay. Is there like a weird echo or? Louder? You got it. So my name is Hoekstra. I, along with several of my colleagues, including Ranjan, am one of the co-founders of the RaDVaC project. RaDVaC stands for Rapid Deployment Vaccine Collaborative, which it kind of embodies our original thinking and our original mission for the project. I should say that we began in the early stages of the pandemic, actually before COVID was sort of registered or acknowledged as a global pandemic. I can recall the sort of founding email was Dr. Preston Estep emailed myself and a couple of our colleagues who had worked together on various projects in the past in, I think, either late February or early March 2020 with a question, right? The question being, does anyone know of a project which is pursuing vaccine access, accelerated vaccine access in a way that conventional vaccinology at that point was not going to be capable of? And we, the group to whom this email went out, largely bioscientists. I myself am a molecular biologist, a geneticist. I had never worked on vaccines in particular, or in specific. My work previously had been in protein design and sort of like pre-drug discovery work. And for all the reasons that proteins make terrible drugs or challenging drugs, they actually make pretty good vaccines because they tend to be fairly immunogenic. So there was a fair amount of crossover, but I didn't know that at that point. So, but we were, as a group, able to kind of look at the transmission dynamics of the SARS-CoV-2 virus at this point, in the earliest days of the pandemic, and extrapolate based on what everyone's sort of prediction was at that point for when vaccines would be accessible. And this is accessible in quotations because accessibility obviously turned into a global challenge, more so, or to a greater extent than a regional one. Accessibility here in the States is great for us. It doesn't solve a long-term problem of gaps in accessibility that that are distributed across the world, especially where there's poor public health infrastructure and just a poor sort of ability to respond to and contain infectious diseases. So, at any rate, we were, the expectation at that point was that vaccines would be accessible in 12 to 18 months. Now, that would be unprecedented for the time. Previously, the record for sort of completion of a vaccine clinical trial from preclinical trials on through phase 3 and commercialization of that vaccine was, I think, 4 years. So 18 months would, again, represent kind of an unprecedented speed. And at the same time, we were looking at the transmission dynamics of this pathogen with the understanding that useless is a strong word, but to say that it would be too little too late, I think encapsulated the mentality and the sort of realization that we had at that point, that when you're coming up against infectious disease, especially in an epidemic, or in this case a pandemic circumstance, time is the enemy. You're dealing with an exponential function. The more quickly you can intervene, especially with immunity, the better, right? The less your outcome will be. So anyway, that's how RaDVaC started. There are a number of us who kind of constitute the core team, and many of these folks are in the Boston area. Most of us knew each other through working in George Church's lab at Harvard Medical School at various points in the past. But as you can see on the map there, we, because of the nature of our project being an open source sort of collaborative initiative, we've sought out investigators and researchers and individuals and corporations and various sort of agents of vaccine development across the world. The image here is, I think, a little bit out of date. I think we have a few more in Australia and maybe distributed across Asia by now, but this is also sort of a self-reported map, right? This is our open directory of people who want to be found, who want to collaborate, who want to develop vaccines. We've also been very fortunate to, I think starting in 2021, after about a year of technology development, we've had some very generous support from people who are interested in and sort of supportive of decentralization overall, and who kind of recognize the importance of rethinking and restructuring global public health infrastructure. So some of this I've already discussed, but as I've mentioned, the distribution of vaccines was certainly not equitable. And this is more than just sort of an ethical issue. It really turns into a practical issue when you're combating an infectious disease, especially with as globalized an ecosystem as we've had. Obviously, the United States did not rank particularly highly in access to vaccines. We still ranked far higher than most countries on Earth. This graphic is a little out of date now, but you could see, as of almost a full 2 years into the pandemic, most countries on Earth did not have good access to vaccines. And I could get into, and maybe we'll get into in the Q&A section, kind of transmission dynamics and sort of the math of pandemics and why it's important to have immunity as opposed to some other sort of non-pharmaceutical interventions, and why it's more protective, sort of more protective mechanism on a population scale than it is on an individual scale. But as you can see, and as has been borne out with what is now history and still in the present, conventional vaccine development did not succeed in containing the virus and containing the disease, and certainly not when you expand your view to a global scale. So again, sort of ethical, economic, and beyond are the consequences of all this poor accessibility. And as Dan mentioned, access was both a technical problem but also a political problem, right? It is sort of a failure of the technology at the time and a failure of the infrastructure that sort of enables vaccine development and vaccine deployment at the time, because it wasn't built for responding to pandemics. It wasn't built for responding to emergencies the way that we had. So the way we've kind of thought about vaccine access is as sort of a healthcare or a pharmaceutical analog to the trickle-down economic theory, where you have a high amount of access in select groups, and hopefully after that you can filter access out to the people and countries and places with poorer access. Unfortunately, this doesn't work. As we've seen, this doesn't work well for a pandemic. It probably isn't a good public health policy. I think it's a policy that's born of sort of short-term thinking and sort of, I don't want to say greed, but very sort of nationalistic thinking that, again, when you're in a pandemic circumstance, it doesn't really work well. So part of this is because there's a lack of infrastructure around the world, and that that takes the shape of not only lack of pipeline from rich nations to poor nations, but lack of sort of production and development infrastructure in poor nations. It was interesting that on something I didn't know before starting on the RaDVaC project was that on the entire continent of Africa, there was virtually no vaccine development capacity whatsoever. Now, various organizations are beginning to try and fix that now. We've seen, the WHO invest in Afrigen, I think was the name of the initiative, to try and pirate Moderna's vaccine and develop it in South Africa. They had mixed success. They reached some technical milestones, but I'm not sure a vaccine ever actually got out the door of their facility. So what we envisioned was, I'll go back for 1 second. What we envisioned was sort of upending this model in kind of what would it be a grassroots sort of way, that we could develop technology that's so accessible that you don't really need the permission of high-income countries. You don't need the permission of the conventional stakeholders in vaccine development. in order to begin doing development of your own. I say you, and you know, I address you as individuals, but really what we were thinking of is sort of the scale of countries and cities, right? That every country and every city should have the ability to participate in the process of vaccine development, or at least deployment, right? At least production and then deployment of vaccines. But there are technical challenges to that we can get into later. But in terms of the limits to access, we encountered what we thought was a really interesting sort of trolley problem, right? This is a problem of rationality in which regulatory stakeholders, of which there are many, right? The FDA is not alone in sort of restricting access over time or you know, over space to vaccines, but the FDA is of course a stakeholder in that. They have regulatory control of the switch, right? And they made a risk assessment, right? These various political agencies and regulators made a risk assessment having to do with, do we permit access to a vaccine that is not 100%, you know, tested, proven, up to the standards of previous vaccines? And they, you know, I think many of us at RaDVaC have had to evolve our kind of thinking about the FDA in a sympathetic way, because there's a mandate of the FDA, of course, to deploy public health tools, and you're dealing with a public that is not completely rational, right? Like, none of us are homo economicus, none of us have completely rational decision making. And obviously there's sort of an anti-vaccine sentiment that's been growing, especially in Western countries for a long time. That kind of sentiment and that sort of you know, impediment doesn't just limit vaccine access for the people that you know, that would otherwise opt to or not to have vaccines, but it really has a much more widespread impact than that, because those people will of course go to work, they will go to school, they will go to their families, they will not be protected, they will not have sort of an ability to protect the people around them that they would have if they were otherwise vaccinated. So anyway, the math here is not quite accurately represented in this graphic. The risk of vaccines is actually staggeringly lower than the risk of the virus itself. And this is true across, I think, every instance of vaccine development, including some of the sort of black spots on, or the black marks on vaccine development across history, where you have, you know, instances where polio vaccine was inadequately inactivated, and you had, you know, instances where there was terrible reactogenicity with you know, children and others who had received vaccines. Still, there is almost, I think it's safe to say, there was no instance of vaccine development that was actually worse than the disease it was trying to protect. And you know, in modern vaccinology, this is even more true, right? We have a clear mechanistic understanding of most of the vaccines that are even in preclinical trials, and we can make sort of informed strategic assessments of their mechanistic risk, right, or the risk from a mechanistic level. Now, that's not completely straightforward, and it's not a guarantee, which is why someone is on the track that is sort of leading away from the world population that's otherwise sort of susceptible to this virus. But the math skews heavily in the direction of, you know, permitting access to vaccines. And of course, oh yeah, so you can see the math here is a little more representative. When you've got 8 billion people who are otherwise susceptible to vaccines, or susceptible to viral infections, and of course the sort of catastrophic wake that had already taken place at that point of thousands and eventually millions of people who were infected and either died or suffered long-term consequences of what's now being called PASC, which is the technical word or technical term for long COVID. And that's, you know, we're beginning to be able to calculate the impact there. But we at RaDVaC, you know, we thought that there was sort of an asymmetric risk assessment in this scenario, right, in the world that was happening at the time, where the risk of the virus was being underestimated by these stakeholders, by the people who would either permit or restrict access to vaccines, and the risk of the vaccine was overestimated. And again, you know, this is not just our opinion. This is sort of statistical and molecular biological, you know, understanding of how vaccines work versus how viruses work. And you know, even with a sort of a modest understanding of how SARS-CoV-2 was operating at that point, or how risky it was at that point, we could see that, you know, the risk of a vaccine was far less than that of letting the virus kind of run wild. But of course, you know, permitting access would of course be an active choice, so you have to take responsibility for that, and I think that's also really challenging for global stakeholders. So what we at RaDVaC sought to do, and what we continue to seek to do, is to improve access to vaccines by improving access to sort of the underlying technology that enables vaccines, which is, you know, sort of the constellation of sciences known as Vaccinology. Deployment is in our name, and deployment is kind of the end goal of all of that we get vaccines out to people, to individuals, to, you know, immune systems, so that they can move through the world with better immunity and better protection. But we also, you know, we acknowledge that upstream of that is, you know, a real technical challenge. And that's kind of where we focused, at least primarily. I think more recently, we've gotten more interested in talking about kind of the regulatory and political and economic framework that pervades vaccinology, and thinking about the ways that we can improve those, you know, sort of tools or impediments, make them less of impediments, make them more sort of more rational and much more resilient tools for doing public health. The way we pursued this, especially at the outset, was through what we kind of considered vaccine developer kits. In software development, SDKs or software developer kits are accessible to software developers around the world, and most of them are you know, fairly easy to pick up and play with. If you're familiar with code, or if you're you know, familiar with that programming language, you could pick it up, play with it, plug into it, develop applications for it. And we really looked at Linux as kind of an interesting, you know, paradigm-changing piece of software or you know, invention that changed the way personal computing became accessible, and ultimately it changed the trajectory of evolution for personal computing. And we thought, well, wouldn't it be interesting if we open sourced and made the technology of making vaccines and ultimately testing vaccines fully transparent, fully open source, but also technically accessible, right? And what I mean by that, you know, is that it has to be designed in such a way that is replicable in very tolerant conditions, or that it's very sort of, you know, you can do this without $1 billion, you can do this without a BSL-3 laboratory, or without ultra cold, you know, vaccine storage. So we started publishing a series of white papers online that detailed our initial designs for an open source vaccine. We simultaneously, as a core team, began actually prototyping these vaccines, or vaccine candidates, not with the intention of commercializing them, not with the intention of sort of developing these fully ourselves, but with the intention of, you know, proving that it was technically possible to do this without $1 billion and without a BSL-3 laboratory, using, you know, mostly ingredients that could be purchased commercially, almost all of which could be purchased sort of off the shelf. But of course, you know, when we published these, we put the invitation out for improvements, right? That we wanted people to play, you know, open these up, play with these, improve upon these. And again, I say people, but what I mean, you know, more ambitiously is researchers, institutions, even companies. Like we have, we worked with Creative Commons to both, you know, employ some existing like Creative Commons licenses. So CC4, I think, was one of the first things that that they began using for open source COVID things. But then eventually they came up with a whole patent structure that was sort of designed to help people, help commercial developers use what would otherwise be patented technology to develop vaccines. And I'll just go back for just a moment and say that you know, throughout the pandemic and still to this day, we continue to see improvements in our white papers. We continue to see improvements in the formulation of our vaccines, in the targeting of our vaccines. We continue to you know, help and be helped by collaborators around the world who are doing some of the hard, molecular bio work and immuno work of selecting better antigens, stabilizing those immunogens, stabilizing the nanoparticles that we use, otherwise improving kind of the technology that enables vaccines and the technology that comprises vaccines. So some of the core components of the technology that we sought out to develop, we were really interested in mucosal delivery and mucosal immunogenicity. That was one of the sort of core principles of our vaccine development. It's not necessarily mandatory in terms of, you could take one of our vaccine candidates, fork it in a direction that it's administrable intramuscularly or orally. I know of some people who were testing it that way. We in the core team were primarily interested in mucosal. I think we're very interested also in oral route of delivery and immunogenicity for respiratory diseases like COVID. Rapid production, of course, that's key in an emergency situation. Safety is also quite important, not just safety in terms of sort of endpoint and reactogenicity, but also safety of the materials themselves. So not working with any sort of self-replicating hazardous materials, not working with any live virus. All of these things would constitute barriers to access upstream of deployment, right? So barriers to access to just the technology itself. We also thought that generalizability was highly important, that our Gen 1 vaccine, which I had in my nose, I think on April 2nd or 3rd of 2020, wasn't very good. It wasn't a very well-designed vaccine. At that point, we were using computational biology to try and predict what good immunogens would be, and then array them on nanoparticles that we could make in a lab for very cheap, and then administer them through the nose in a nasal sprayer. Those sort of computationally predicted immunogens were not very good, right? They were not very accurate in terms of what actually turned out to be good immunogens that constituted better vaccines. But the generalizability of the technology that we were working with, and we've been developing since this point, the benefit there, and the benefit of the structure of our approach as opposed to a commercial approach where they're kind of locked in, was that we could evolve the vaccines as we gained a better understanding of the virus, right? So as better understanding of antibodies, better understanding of T cells, better understanding of how our immune system responds well or responds most effectively to these targets, we could include those targets, right? We could include better and more stabilized targets. So, we had a list of criteria there as well for immunogen selection. We wanted things that would probably not mutate away, right? And there we could look back on the phylogenetic history of coronaviruses and see which protein structures, which antigens are most stable versus which ones are most mutable, which ones were likely to elicit a B cell response and ultimately kind of an antibody response, versus which ones were likely to elicit a T cell response. And from there, you could differentiate different T cell responses. CD4 versus CD8, for instance, was something that we looked very hard at. And then, again, through the ability to improve and through this sort of distributed ecosystem of developers and scientists co-working on this project, and not all of them were co-working. Some of them took it and ran in their own direction, and that's fine, right? Like, our job here was essentially to be a catalyst. And I think, there's a nuance to that got lost many times in many conversations that we had. People expected us to be a pharmaceutical company, right? People expected us to run clinical trials and publish, clinical trial data. I was torn, right? And I think many of us in RaDVaC were torn between sharing what data we could collect on a very small, small study, small N studies that were very independent, and, we didn't necessarily want to influence people to self-experiment. I think there's a moral gray area there that we can't make promises. We shouldn't make promises. There's also a legal gray area there that we shouldn't make promises, and we didn't want to step on the toes of the FDA. But that we should just publish information, right? We should publish sort of the raw technical information of vaccinology and hope that it inspires others to pick that work up, to iterate that work, to evolve that work, and potentially to collaborate on that work. And we saw a fair amount of collaboration. We got tons of emails from researchers around the world who said, I think you could improve the design by doing this, right? You could improve the design by adding this peptide sequence. And we did. We tried a bunch of things. And we published, we continued to update our white papers. One other note here, self-administration I think is a really undervalued sort of asset of mucosal delivery. Vaccine administration is a really sort of, a real challenge in public health because, there's a saying that vaccines are really only as good as vaccinations, which is to say that they have to be deployed, they have to be administered in order for them to be useful. You can have the best vaccine in the world, and if people don't go to the clinic or don't go and get the vaccine, it's not going to have a particularly strong impact on not only those individuals, but as a population. So anyway, these are some of the design criteria that we had in mind. We've continued to kind of survey the landscape of technologies around vaccine development. You can see across the top columns there, or the top row, those columns are kind of distributed or broken into different technologies for vaccines, different platforms for vaccine development. And then along the first column there, you can see the rows are attributes of those technologies. And we, not the most quantified sort of rank system here, but we've continued to, both as a core team and as a global community, continue to kind of look at what technologies are most useful or what the benefits and drawbacks are of various technologies for vaccine development. I think we started and even now we kind of maintain an agnostic approach to what is the best vaccine, right, or what is the best vaccine technology. There are pros and cons of each one. And our goal, I think, is and has been to increase access to any useful vaccine technology. We started with peptides. We started with designing custom peptides that matched antigenic sites on the virus, and then arraying those peptides on and in nanoparticles that are made of a derivative of chitin. So the derivative is called chitosan. It's very cheap, it's very abundant, it's very tolerant under sort of various storage conditions. It's very easy to make. They actually self-assemble into nice little nanoparticles, and you can suspend that in a solution and spray it up the nose very easily. But we've also been really interested in, of course, RNA and various formats of RNA, and even DNA vaccines. And we want people to make those technologies more accessible. We as a group want to make those technologies more accessible as well, because there are still technical limitations to what those vaccine platforms can do right now. And part of the reason for that is because they're in such walled gardens. And the walls there are billions of dollars high. And we think if we can bring down those walls a little bit in terms of access in both the financial sense and in an information sense, that we could maybe catalyze better improvement, better innovation, and hopefully a renaissance in better sort of biosecurity tools through whatever platform works best. One point at the bottom there, clinical trials were, as Dan mentioned, probably the longest component of vaccine deployment, right, and the largest and longest hurdle in the way of vaccine deployment. Part of this is regulatory sort of tradition or convention, and there's sort of a single global gold standard or a single national gold standard for what constitutes a good clinical trial for vaccines. Unfortunately, the randomized placebo-controlled double-blind study of vaccines requires that half your study population be subjected to, a pandemic without any protection. There's ethical concerns about that, rightly so. So we, as a group, many of us were interested in developing better clinical trial designs. And so we published kind of the framework for a clinical trial design that was open source. And we hope that people would pick it up, consider it. It's quite different from the randomized placebo-controlled double-blind vaccine clinical trial that is sort of the current gold standard. It involves a two-stage challenge trial where you have, and this, I should say that this design, this clinical trial design, is particularly effective or particularly useful. It's geared towards multivalent or pan pathogen family, right? So pan coronavirus or pan influenza virus vaccines, which of course we and others have been interested in, but we've been able to pursue with a lot of flexibility and a lot of speed, because we're not sort of, we're not beholden to the same regulatory framework that commercial developers are. We're just doing science. So anyway, if you're interested in vaccine clinical trial design, I think please check it out, and please help improve it, right? Because it's not, there's no final version of this. Because it's open source, you can edit it, right? You can propose edits, you can download it and rewrite it, and if you think you have a better idea than us, I definitely want to hear it. Just touching on sort of the impact of being open, right? The impact of operating in an open source way. That we sought to improve access across a number of dimensions: participation, quality, speed, equity, trust, effectiveness. Each one of these are non-trivial in terms of public health, right? Each one of these is non-trivial in terms of actually getting vaccines into vaccination form, right? Into deployed form. And especially on a global scale, right, where you have, right now, very few humans on Earth have the ability to participate in vaccine development. We want to improve that, right? We want to drive that access up so that we can improve, equity. And again, equity in a pandemic sense and equity in a vaccine sense is not just a moral issue. Of course, it is that, but it's more, right? It is a practical, practical obstacle that has to be hurdled. Trust, public trust. I think there was a broad misconception of our project as being kind of a DIY vaccine. I think that's a really interesting and probably, I would say it's a mischaracterization. I think it's an interesting characterization to say that a highly accessible open source vaccine would be kind of DIY. I think that the point is that it's highly transparent and highly accessible, so that not so that everyone can be making vaccines in their own bathtub, but so that every country on Earth has the ability to not fend for itself necessarily, but to be self-sufficient, right? To be disintermediated from the trickle-down model of vaccinology right now. So all of these are dimensions that I think are important, that we think are important for enhancing global health. So again, Linux was an interesting model for us to kind of borrow from philosophically and practically, because it pervades digital technology and computer technology today, whether people know it or not, right? Linux kernels drive most of web infrastructure. They enable most of web infrastructure, and it's not because they're free. It's because, well, not because they're gratis free. It's because they're libre free, right? It's because they've had a chance to be developed in an open source capacity, in an open source framework where people have been bug hunting and optimizing and sort of re-engineering and redeveloping that code for so long that it's very resilient, right? It's fairly robust, and of course bugs have been found. Bugs will always be found, and hopefully in an open source ecosystem, people have the ability to find those bugs very quickly and respond to those bugs quite quickly. So we've envisioned a similar sort of framework for vaccine development or for biosecurity. I've just added a slide here on some remaining technical challenges. These are things that we're actively thinking about and working on right now. There's a problem in the development of public goods, which is not unique to vaccines, but of course unique to lots and lots of other public goods around the world. But how do you incentivize production, right? How do you incentivize sort of the investment that vaccine developers have to make in an open source product, right, or an open source framework? We don't have a good answer to that, right? We thought, and we still think, that a pandemic should be incentive enough, that companies and researchers and universities and public health institutions should be encouraged and motivated merely by the sort of stakes of the infectious disease to invest in open source infrastructure and open source tools like Vaccinology. But we haven't found a good way to really sort of incentivize that in a capital-driven way, right? Those two things don't quite fit together yet. So again, if you have ideas, let's talk about it. I love hearing ideas about this, and there's a lot of people working on this for other public goods. We've had interesting conversations with Vitalik Buterin about the way that cryptocurrencies could potentially enable public goods development and incentivize public goods development in a sustainable way. But at any rate, it's something that we're working on and thinking about, and I'd like for more people to be thinking about it too. The one in the center there is vaccine factories in a tube, and this is kind of the what I would consider, what many of us at RaDVaC would consider kind of the holy grail of enabling capacity, right, or building capacity on a local scale across the world, right, that you're not just shipping vaccine, but you're actually shipping the ability to produce vaccine. So the ideal vaccine factory in a tube is, of course, thermostable, right? You can ship it at room temperature. You don't need ultra-cold storage, that it's shelf-stable, that it's sort of minimally resource demanding as possible, that it's kind of a just add water model. And there are some really interesting technologies that might come together to enable this kind of vaccine factory in a tube technology. We are beginning to invest in those technologies, right? We're beginning to invest in things like cell-free protein synthesis. A lot of people have been interested in approaching this kind of technology from different angles, and I think all of them are interesting, all of them are worth considering. Cell-free protein synthesis, I think, is one of the most interesting and maybe top of our agenda there. Also, maybe making open source, making open source nanoparticles that carry vaccines, right? That carry immunogens and can deliver the sort of immunological payload of vaccines. So, in mRNA vaccines, for instance, lipid nanoparticles are still quite proprietary. They're not actually patented. They're just trade secrets. And the sort of chemistry of how to produce those nanoparticles is not well distributed, right? Or the knowledge of how to produce those nanoparticles is not well distributed. I think that if we come up with an open source model that we can improve the efficacy of those delivery vehicles, we can improve the immunogenicity, we can sort of drive down the dose that is required, we can drive up the tolerance of storage conditions. So that's something that we're very interested in as well. I think I'm probably well over time, which is funny because it's only like 12 slides, but I could talk about this for a really long time, and Ranjan can talk about this for a really long time. I'm happy to talk about it for as long as you want to stick around. But yeah, if anyone has any questions at this point, I think that's the end. That's my last slide. So thank you. Again, I'm Alex Hoekstra, and thank you on behalf of RaDVaC.
See if we can switch the